You probably know what DNA is. We've talked about that, and you've probably heard about chromosomes before. But now what we want to do is sort of link those ideas together and how you go from DNA to what a chromosome is, and the relationship between those. So what happens is a cell, in a cell like yours, for example, the DNA is very, very, very long. And in order to sort of contain it, what happens is it is sort of, it's wrapped up. It's wrapped up and it's maintained in pieces or strands. And so what happens is the DNA in a cell like yours is wrapped around these proteins, and then those proteins, so here's one of your strands of DNA, for example, and you have many strands of it, but here's a strand of your DNA, and it'll be wrapped around these proteins, and then those proteins are wrapped around and form this almost rope-like structure, and that's called chromatin. And normally when your cells are just not dividing, they're just simply going about their daily cell life, what you would have in the cell is really chromatin. So you wouldn't actually see chromosomes unless the cell was dividing. So what happens is normally a cell is found in its sort of chromatin state, and then when it gets ready to replicate, what will happen is that chromatin will wind up even tighter and form these chromosomes. Now the tricky part is that when you first see a chromosome during the cell cycle when it's dividing, what you actually see is a chromosome that's duplicated, meaning that it's already gone through DNA replication and it's actually has double the amount of information, everything's copied in it. And so you'll see a thing like this. So we'll talk about this a little bit here. So here, once again, here's my DNA, here's my proteins that I wind that up into chromatin, and then here's my duplicated chromosome. And so what happens is that chromosome then, that duplicated chromosome, has two parts to it typically, and those two parts are called sister chromatids. So here is a duplicated chromosome and it has two separate parts. They're called sister chromatids, and they both contain the same genetic information unless there was an error when they were copied. Okay? Those two sister chromatids then are attached, and they're attached sometimes, in the middle or near the end. They can be attached in different places, and that attachment site where the two sister chromatids are connected is called the centromere. So that's where the two sister chromatids are connected. What this picture is here, this is called a karyotype. A karyotype with a K is a picture of all of your chromosomes. And so what ends up happening here is that you have, basically, assuming everything was done correctly, you should have two of every chromosome. You got one set from mom and you got one set from your dad. Okay, so for example, here's your chromosome 1s, and so here's one pair, and then here's the other pair. And we say that the two chromosome 1s are homologous chromosomes. And what that means is that they carry the same type of genetic information. Not the same exact genetic information, but the same type. So if on one chromosome, for example, down here, let's say there's a piece of DNA there and that DNA codes for eye color. That means on your other homologous chromosome at that same relative spot, you should have information that also codes for eye color. This one, though, might have a code that codes for brown eyes. This might have a code that codes for blue eyes, but it carries the same type of genetic information in the same relative spot. Okay, so you have your chromosome 1s and 2s and 3s, and those are all what we call homologous chromosomes. If you are a male and you have an X chromosome and a Y chromosome, those are called your sex chromosomes. Those chromosomes are not homologous because you'll notice that the Y is quite different than the X. The Y is a very small chromosome and has relatively few genes on it compared to the X. All right, so what's going to end up happening then is the cell you're going to take all the information in the cell, and there are two sort of separate processes that can happen here. One is you can take a cell and you can copy all the information in a cell, and then you can divide that cell into two cells that are identical basically to the first starting cell. And what that results in is what we call diploid cells. A diploid cell is a cell that has two sets of chromosomes in it. So you notice we use this 2n. N equals how many chromosomes are in a set. Now, in a human, there are 23 chromosomes in one set. Okay? In different animals and plants, the number in a set can vary tremendously. But in the case of a human, the number of chromosomes in one set is 23. 22 are what we call autosomes, chromosomes 1, 2, 3, and so forth. And then one pair of them are the sex chromosomes. So 23 in a set. Okay, so a human, if you were to take, say, a typical cell from the heart of a human or the brain of a human or a skin cell from a human, and you were to look and count how many chromosomes it had in it, assuming you could do that accurately, it would have 46 chromosomes, because it would typically be a diploid cell and it would have 2n, and there's 23 in a set, so 23 times 2 gives you 46. If you were to take the haploid cells, which would be a sperm or an egg, you would have a haploid cell, you would have an n value, in this case of 23. So once again, you get half of your chromosomes from your mom, half from your dad. 23 plus 23, 23 in a sperm, 23 in an egg, you get those together, that gives you a total of 46. So the only cells that typically would be haploid in a human would be the sperm and the egg. So that process we'll talk about later on how to make those is called meiosis, where the process where you copy everything and make two diploid cells is going to be called mitosis, and that's what we're going to get into next.